Well, just moderated today. Had a cold all weekend. Wife had surgery this morning. Got her knee replaced. Just got called. Everything went well. She'll be out of the recovery room in another hour or so. But my brain is just kind of not functioning at its high speed, which never is too fast. What we're going to do today is talk about lipid metabolism and distribution in our body. So in GI physiology, the reason we eat is reason we eat is to get nutrients to the body and make ATP and to survive by doing battle with the The three main food groups that we can get energy from are lipids, carbohydrates, and proteins. And of those three, we'll cover carbohydrates and proteins in another lecture. That's relatively simple compared to lipids. So we're covering the more convoluted thing in an aspect first. So we'll start off with looking at this, how we're getting lipids into our body to serve as, well, the many functions that lipids serve for us. So first question that comes to mind, well, just how much are we talking about in terms of lipids that we're ingesting? It's pretty variable, 60 to 150 grams of lipids per day. I probably exceed that 150 on occasion. But that's about how much our GI tract has to cover. Now, there are lots of different lipids. So what do we have in terms of dietary lipids? 90% of all the lipids that we eat are in the form of triglycerides. So when I think of dietary lipids, I primarily am picturing triglycerides. You remember, talk about triglycerides in Foundations of Life Science. That they're a great way to store energy, because if you were given a pound of fat and a pound of glycogen, there's a lot more energy in the pound of fat because it's 9 kcals per gram, but also if you squeeze the pound of fat, all you get really is fat oozing between your fingers. Isn't that a pretty thought right after lunch? If you squeeze that pound of glycogen, you'd see a lot of water coming out, so glycogen is highly hydrated. So 90% of what we eat, dietary lipid, is triglyceride. What other lipids do we eat? Well, that remaining 10% is primarily made up of cholesterol and cholesterol esters. Cholesterol ester is just cholesterol with a fatty acid bound to it. Phospholipids, because we're eating cells, we have phospholipids in those membranes. And a really small amount is actually free fatty acids. So we don't eat a lot of fatty acids. They're primarily coming to us attached to other things. So you put those in your mouth. What happens? How do we start digesting? The very first part of digestion occurs with saliva. And in saliva you can find something called lingual lipase. Lipase indicating it's an enzyme that is involved in metabolism of lipids. So we find lingual lipase in saliva. The trick here is lingual lipase isn't active at neutral pH. Its pH optimum is acidic. So as we're eating lipids today as adults, the lingual lipase isn't doing much until it gets to the stomach and there's an acidic environment. So we can get along fine without lingual lipase. Okay? It's different. It's not, babies can still get along with that. But for babies, it's a little bit different. So lingual lipase, also called acid stable lipase. Come back to that in just a moment. So when you're ingesting now, there's very little change in the lipid, no chemical digestion taking place. Now we get into the stomach, And in the stomach, the lingual lipase can get activated. The stomach also secretes a lipase, and it's called gastric lipase, which is nice enough. It tells us where it's coming from. So we have another enzyme, a second enzyme for digesting dietary lipids. This is gastric lipase, and once again we see gastric lipase isn't really active in the stomach because it has a pH optimum at around neutrality. So the gastric lipase isn't going to activate until we get into the duodenum. And the pancreatic bicarbonate neutralizes that, the count. Gastric lipase, when it's active, acts on triglycerides with short and medium length fatty acids. But this, the lingual lipase, gastric lipase, I'm sorry. Gastric lipase in the adult doesn't activate until we get into the duodenum. Gastric lipase can break apart triglycerides, but it primarily is active on triglycerides with short to medium length fatty acids attached to the glycerol backbone. Well, where do you find that? You find milk fats have a lot of short and medium length fatty acids. So gastric lipase is going to be particularly good at digesting milk fats, but it's only active near neutral pH. And the reason for bringing that up is that note down at the bottom, even gastric lipase doesn't accomplish a whole lot of digestion in us as adults. But when is the one time that we have a lot, where milk, and milk fats would represent a high percentage of our dietary lipid, it's when milk is a primary ingredient in our meals or nutrition, and that happens with babies. The other thing you see in babies is their stomachs have not yet developed a great ability to secrete hydrochloric acid. So gastric lipase becomes a more significant enzyme for digesting lipids when in babies, where it can be active in the stomach because the stomach isn't so acidic. Right.
Is lactose intolerance just about lactase, or potentially also a deficiency in this gastric lipase?
No,
Just lactase.
Lactase.
Okay.
The carbohydrates. Okay, so I just thought that was kind of cool. Bottom line here, as adults, we get into the duodenum and very little has been accomplished in terms of digesting dietary lipids. Okay, another reason. So why is that? Reason number one, the enzymes necessary. Lingual lipase doesn't work much anyway, but it's active in the stomach in an adult. Gastric lipase in the adult doesn't get activated until the stomach, but probably the biggest reason, a major reason we have very little lipid digestion until the duodenum in the adult is we haven't emulsified the fat. So if you've anybody ever cooked pasta, boiled pasta in a pot, okay? You ever put like olive oil, put some olive oil in the pot when you're boiling your pasta? Sometimes people do, sometimes people don't. If you've ever done that, if you ever poured oil into water, you'll see as it's heating up that it's just kind of floating around together in one large clump. Well, that's what's happening to your dietary lipids until they get into the duodenum. They're going to clump together because they're hydrophobic and largely hydrophobic. So what we need to do when we get in the small intestine is emulsify them, break them into smaller droplets, little globules, to increase the surface area to volume ratio. That happens fairly early in the duodenum because of the release of the bile salts that we talked about last week by by the liver, concentrated in the gallbladder and then released by by the gallbladder due to the presence of what? No, bile will be released from the gallbladder in response to cholecystokinin. Okay, so we get the bile salts released, they emulsify. Instead of large globs, we have smaller globules. That increases the surface area, which is where the enzymes can act. We said the other day that what are bile salts? Either taurine or glycine attached to bile acid, and that's cholesterol based. So you get glycocolate, taurocholate. Those are the two primary bile salts. And you just look for words that look like chocolate. You know what people are talking about. Okay. Two other words to recognize. Bile acids. Here are two names for bile acids. Cholic acid. Notice that cholic acid, because it's a weak acid, you could also refer to it as the salt of that acid, and that's cholate. That word looks a lot like chocolate. And then chenodeoxycholate, or chenodeoxycholic acid. So those are two examples of bile acids, right?
In the case of a gallbladder removal, which is relatively common, is there such a thing as exogenous administration of order wouldn't survive?
Well, usually the person is still making bile. They're just not storing and concentrating. And when they eat, there will be a signal, the release of secretin to tell the liver to make more bile. So what you would do typically is just have the person eat a low-fat diet so that they can handle that reduced load with a reduced amount of bile salt. If they didn't do it, somebody has lost their gallbladder, eats a high-fat diet, what are they going to end up with? Huh? A big word beginning with S. Steatorrhea. Right? So greasy, smelly stool. So it probably doesn't take long before they decide, no, I'm going to go down and reduce the fat in my diet. Okay, bile salts made by the liver. The liver is recycling a lot of that, so the liver converts cholesterol to bile acid. Bile acid gets the taurine or glycine attached to it, forming the bile salt. Bile salt is secreted by the liver, concentrated in the gallbladder, released by the gallbladder in the response to cholecystokinin, and in the intestine, we would find both a combination of bile salts and bile acids. If we were looking at bile and what the liver was secreting, it's almost all bile salts. So there's a conversion of bile salts to bile acids in the intestine, and later in the intestine, these get reabsorbed, taken back by the hepatic portal system, and about 95% of all the bile salts released by the liver get returned to the liver, either as bile salts or bile acids, and used to remake new bile salts over and over and over again. Okay? So now, what happens in the small intestine? We've emulsified the lipid. Now we need to break it down chemically. So we have emulsified triglycerides, okay, 90% of all the dietary lipids. We have emulsified cholesterol esters, emulsified phospholipids. We need each of these molecules is too big to pass directly into the body. So the first thing we need to do is break them down into small enough pieces which can then get either diffuse into the body or be transported into the body. And that chemical digestion is accomplished by pancreatic enzymes that are getting dumped into the duodenum. So here are the different types of lipids, primary types of lipids we find in the diet, and how we digest them. So at the top, we have catabolism of triglycerides, 90% of what we got. Then catabolism of cholesterol esters and catabolism of phospholipids. So if we start at the top, triglyceride or triacylglycerol, triacyl meaning three acyl groups, fatty acids bound to a glycerol backbone. First thing that we're going to do is we're going to take pancreatic lipase. Remember, this is 90% of all the lipid in our diet. So pancreatic lipase is going to be responsible for breaking down 90% of your dietary lipids. And what it does is it takes those fatty acids and clips off fatty acid from the number 1 carbon and the number 3 carbon of the triglyceride. And the products you get from the action of the pancreatic lipase then are going to get 2 free fatty acids. The one that came off of carbon number 1, the one that came off of carbon number 2. And then the remaining structure is glycerol with a fatty acid still bound to carbon number 2. So the official name for that is it's monoacyl glycerol, one fatty acid bound to glycerol, and it's bound to the number 2 carbon. So the official name is 2-monoacyl glycerol, where the 2 is indicating that we're bound to carbon number 2 in the glycerol. Okay? And these are small enough and water soluble, lipid soluble enough that we can absorb them. So pancreatic lipase, end of story for triglyceride. Cholesterol esters. I said cholesterol esters are cholesterol with a fatty acid bound to it. And while this is really hydrophilic, it's just too big to get in or diffuse into the cell. So what we need to do, the only thing we have to do here is clip off the fatty acid from the cholesterol. And the enzyme that does that is pancreatic cholesterol esterase. So it clips the ester bond binding the fatty acid to the cholesterol. And pancreatic cholesterol esterase then yields cholesterol and 1 free fatty acid. And finally, we've got to digest the phospholipids. And I put in a note here just to take me back to FLS. What's the most abundant of the phospholipids that we find in membranes? Phosphatidylcholine, which goes by what other name? Lecithin. Okay, so here's our example. We're going to take a phosphatidylcholine, which is a glycerol, with fatty acids bound to carbon 1 and carbon 2. And on carbon 3, we have a phosphate, and then subsequently also bound to a cholesterol, to a choline. So the first thing we do is go in and we clip off the fatty acid bound to carbon number 2. And the enzyme that does that is phospholipase A2. And that phospholipase A2 gets activated by the presence of trypsin. Remember what we said about peptidases, proteolytic enzymes, that it's dangerous to make these for the cell that makes them because it could start digesting its own proteins and destroy itself. That's why acute pancreatitis is deadly. If you're going to have phospholipase A2 start to digest the primary lipid in your membrane, that's also pretty dangerous. So what I like to think of here is phospholipase A2 is secreted, but it doesn't get activated until it comes into contact with trypsin, far away from the enzymes that were activated. But the phospholipase A2 does, clips off one of those fatty acids, and what we're left with is a glycerol with 1 fatty acid, a phosphate and a choline, and that's called a lysophospholipid. So a phospholipid like phosphatidylcholine missing one of its fatty acids is a lysophospholipid, and the enzyme called lysophospholipase clips off that second fatty acid. So it's the action of phospholipase A2 and lysophospholipase that yields 2 fatty acids and this remaining structure which is the water-soluble component of the phospholipid, and that's the glycerol backbone of the phosphate and the choline. It's called glycerol phosphorylcholine. That's going to be transported into our body. It's not going to diffuse across a lipid bilayer. Okay, so what we've accomplished is breaking things down in a way, the different types of dietary lipids, breaking them down in a way that can either diffuse into our body or be transported into our body. And remember, our body begins with the luminal membrane of these enterocytes. Okay, so to get it into our body, they just have to cross one membrane. That's the cell membrane, the luminal portion of the enterocyte. Now, when we combine, and just terminology that's going to run rampant here in just a little bit, when we combine bile salts and dietary lipids, what we got was a structure that's in the lumen of the small intestine. Okay? And that structure is called a micelle. And if we have a typical diet, a micelle consists of a lot of triglyceride, a little cholesterol, and bile salts, such as glycocholine or taurocholate. Okay, so we've got the recipe. If you were given in a test question, whether from me or from national boards, and say, where would you find a lot of triglyceride, a little cholesterol, and bile salt? The answer you're looking for is the micelle. Micelles do not exist inside our body. They're outside our body. They're in that tube, in the GI tract. Okay, so we've taken the micelle, which are nice small little globules. We've gone in, our pancreatic enzymes have been breaking down the lipids, and we see they're entering or being absorbed into our body. Okay? Primarily in jejunal mucosal cells or enterocytes. So this is later in the GI tract. Takes longer to digest fat than anything else that we eat. Right.
Micelles never circulate in the blood?
Nope.
Okay.
It's correct. Your statement's correct. Okay, so what do we find inside these jejunal cells? We find a bunch of fatty acids, free fatty acids. We find cholesterol. We find monoacyl glycerol. And we'd also find some of that glycerol phosphorylcholine. But primarily we're going to see a lot of free fatty acids and a lot of 2-monoacyl glycerols. We'll also see free cholesterol coming into the body. So those three things that I have listed on the right-hand side there, the primary products of lipid digestion we're going to see. Now what happens now that we've got them into the body? Well, the first thing I might think of is, well, it would be nice if we just have them diffuse right out the other side, get into the interstitial fluid, and from there into the blood. But that's not the way it works for lipids. It's the way it works for simple sugars resulting from carbohydrate metabolism or digestion. It's the way that things work for amino acids resulting from protein digestion. But lipids take a convoluted route. So once we get the free fatty acids, the monoacyl glycerol, and cholesterol, what are we going to do with them? The first thing we do, especially with the fatty acids, those represent, energy-wise, the most valuable thing that we get from our diet. The first thing we do, once the fatty acid diffuses into the cell, is we trap it. Just like cells trap glucose using what enzyme? Hexokinase. Just like you saw in cells, when they got glucose, they trapped it immediately using hexokinase by adding a phosphate. In this case, we're going to trap the fatty acids and make them more reactive. These are free fatty acids. Now we're going to need to convince them to become, once again, a part of a bigger molecule. So we're going to trap it by adding coenzyme A. We're going to take a coenzyme A and a fatty acid. It's going to require us to hydrolyze an ATP. That's okay. We're going to get a lot more energy than one ATP out of this fatty acid. And the enzyme that's going to do that, to bind the fatty acid to the coenzyme A, is called fatty acyl-CoA synthase. Name tells you what it's doing. It's making fatty acyl-CoA. Well, what exactly is fatty acyl-CoA? It's a generic word. It means fatty. The acyl means acid, bound to coenzyme A. So these enzymes, the family of enzymes, can take different lengths of fatty acids. Can you tell me the name of a specific fatty acid? Omega-3 is a group of fatty acids. Huh? Linoleic acid. Tell me another one. Oleic acid. Tell me another one. Palmitic acid. Linoleic, linolenic, arachidonic acids. Those are all polyunsaturated fatty acids used in the synthesis of prostaglandins, thromboxanes, and leukotrienes. You don't need to know that. Okay? Those are all polyunsaturated fatty acids. Oleic acid is a monounsaturated fatty acid. And if we want to go with saturated fatty acids, I'd remember palmitic acid or palmitate, and also stearic acid or stearate, which is 18 carbons. Okay? There's lots of other ones, but these are just a reminder of some of them that you probably should know.
What's the alternate name, the TH something?
Oh, the alternative name, or another name you see here, is called thiokinase. And thio refers to sulfur. And why would it be called thiokinase? Well, ATP is involved, and coenzyme A, its reactive point, is a sulfhydryl group. So, but I usually go with fatty acyl-CoA synthase. Tells me what it's doing. So first thing we're going to do with those fatty acids is trap them. And as we're doing this over and over and over again, because all the fatty acids are moving in, diffusing in from the micelle, what do we do with them next? Well, we're going to take two of those fatty acyl-CoAs, and we're going to take a single 2-monoacylglycerol, and we're going to, these fatty acyl-CoAs are reactive, and so we're going to stick a fatty acid on the number 1 carbon and on the number 3 carbon of the monoacylglycerol. And the enzyme family that does that are called acyl transferases. We're transferring a fatty acid, an acyl group, from the fatty acyl-CoA onto the monoacylglycerol, forming a covalent bond. So we do that twice, and what we end up with is a regenerated triglyceride, or triacylglycerol, and then 2 coenzyme A's that can be reused for the next fatty acids that are coming along. Where is this happening? It's happening inside the enterocyte, inside the jejunal mucosal cell. Inside of it. Remember, we had to break these down because they were too large to move across the cell membrane and get into the body. So we broke them down, we absorbed them, and then we rebuilt them inside the enterocyte. Okay, and a note about those acyl transferases. I said it was a family of enzymes. Different ones have different kind of specialties. Some are good at handling long-chain fatty acids, others short-chain fatty, well, not so much short-chain fatty acids. But different size fatty acids, different specialties for the acyl transferases. One thing that we see doesn't tend to happen are the short chain fatty acids. Where might you find short chain fatty acids? Pop quiz.
Some member?
No? What might you eat to find short chain fatty acids being produced? Milk, right? Milk fats have short and medium length fatty acid chains. So if we digested milk fats in particular, that's the example I hold in my mind, those short chain fatty acids don't tend to get used to rebuild triglycerides. Short chain fatty acids that get absorbed tend to go diffuse out into the blood, into the hepatic portal system. But the longer, the medium and longer chain fatty acids, that's where we see the acyltransferases working to put them into triglycerides. If you have a short chain length fatty acid, then if it diffuses into the blood, how does it feel about being in the blood? Would you be happy or not so happy if you were fatty acid and you found yourself in the blood?
Is it attached to albumin? Or just
Well, initially it's coming in as a fatty acid, but it's hydrophobic. It's in the aqueous environment, so it's not going to be. It's going to be at a higher energy state. It's going to want to try and get out of there, but we can stabilize it by binding to albumin. So a lot of short chain fatty acids can be distributed by binding to albumin. If I take it all the way back to thinking about, the baby eating primarily milk, it's going to get its fatty acids into the circulation a little bit more quickly than we do. So here we have a fancy diagram. We broke off fatty acids from carbon 1 and 3, have one fatty acid attached to carbon number 2, so that was the 2-monoacylglycerol. That got brought into the intestinal mucosal cell. We have acyltransferase adding a fatty acid to carbon number 1, another acyltransferase adding fatty acid to carbon number 2, so we've regenerated the triglycerides. Where were those fatty acyl-CoAs coming from? We absorbed the fatty acids, and then we use fatty acyl-CoA synthetase, which is going to add the coenzyme A, trapping it and making it reactive to the acyltransferase. The other thing that happens is the cholesterol that we absorb is substrate for another acyltransferase, which can come in and add a fatty acid onto the cholesterol, thereby regenerating cholesterol ester. And what happens now is in that mucosal cell, we take, remember 90% of all our liquid is triglycerides. So we take this abundance of dietary triglycerides that we have regenerated inside the enterocyte. And we've got an accumulating amount of cholesterol esters and a little bit of cholesterol without the ester attached to it. I just use the term cholesterol to refer to both cholesterol and cholesterol esters. But all of these things are accumulating in your enterocytes after you eat a meal that contains fat. Now that's good news for the enterocyte. It's going to have lots of energy, but that's not its job. Its job is to distribute what it's absorbing so that the rest of the body can be fed. And the way the enterocyte does this is a little bit problematic because now I've rebuilt these things that were too bad and too big to get in, well, they're too big to get out. That's the example of NCIS and Jethro building a ship down in his basement. So how do you get it out once you've gotten it built? I nearly fell prey to that one time. I made furniture as a hobby, And so I made a bunch of furniture in my house. I ran out of space. So I started making furniture. Somebody else wanted something, and just tell them, they'll buy the stuff and I'll make it. And so friends of mine down in Washington, D.C. wanted an armoire. So I drew up plans for an armoire, asked them if they liked that, and they said, yeah. I started building it, and it was big. The base of the armoire was about, well, maybe this long and about that deep, and about this tall. So about a little than 2.5 of these panels here. And I very carefully measured out the dimensions of what I drew up, and even went to the point of building a cardboard space occupying model of it, because my workshop is down in my basement. And in order to get anything I make in my basement out of there, I have to take it up a fairly narrow flight of stairs and then have it turn in a corner immediately to take it out the side of the house. I really was proud of myself that I remembered to make sure I could get that thing out of my basement. So you know where it is today? It's in my basement. Because when my friends Barb and Bob came to look at the process and what was happening, they looked at it and they pointed out something I hadn't thought of. It wasn't enough that I could get it out of my basement. They had to get it up their stairs and into their bedroom. And they immediately said, that will never work. So they said, well, try putting these boxes together and see if you can. You'd be surprised sometimes. They took it back to Washington, D.C., and no way it would fit. So now I've got this pretty nice looking bench top for support for my bench top down in my basement. Had it there for 10 years or more. Okay, so this is what the enterocytes are doing too. They're building these things. They're too big to get out. So what they do is they take these triglycerides, they take the cholesterol esters and a little bit of phospholipid, and they combine them with apoprotein. And specifically, apoprotein there's again a family of apoproteins that we're going to be learning about this afternoon. This is our first one. It's called apoprotein B48, or just ApoB48. And apoproteins are important because they can interact with water. So I like the picture of what's produced is an abundance of triglycerides, some cholesterol esters, and then this layer covering of apoprotein B48. Can you think of any kind of food that has a bunch of lipid in the middle, but then something that interacts well with water on the outside? M&Ms. To this day, when I picture this structure, it's like an M&M. Okay? I don't know, digestion just gets me to thinking about chocolate. Okay? This structure is called the chylomicron. So it's another structure. We find this structure is in our body. It's produced by enterocytes. And it contains a lot of triglycerides, which are dietary triglycerides. Triglycerides we just brought into our body. Some cholesterol and apoprotein B48. The moment you see ApoB48, you know it's got something to do with chylomicrons. The moment you see bile salts, you know it's got something to do with micelles. Okay? So now we've taken and made this really big structure. And what we do is exocytosis. The enterocyte exocytoses this, and another key kind of left-hand turn we take. It gets dumped into the lymphatic system. So dietary lipids initially enter our lymphatic system and not the circulation. From there, they eventually drain into the circulation, but not immediately. Okay? So a chylomicron, apoprotein B48, a lot of triglyceride, a little cholesterol, and also we got those phospholipids inserting into the ApoB48 candy cover.
And what's the difference between a chylomicron and micelle?
Chylomicron, both of them, the first thing is what's similar to them. They both contain dietary lipids. A micelle is outside of our body, a chylomicron is inside of our body. A micelle has bile salts, and a chylomicron has apoprotein B48. Yeah.
How do you mean outside of our body?
The lumen of our digestive tract is outside of our body. That we can, if we stretched it out, remember, clean it out, stretch it out, just remove from the mouth to the anus, outside. We can look all the way through it, never had to pass the cell membrane. Okay?
Yeah, no, I get it.
It's not in our body. Same thing as air that is in our bronchi or our alveoli is not in our body. Okay. Same thing as when we talked about the kidneys next fall. When we filter plasma, if you look inside the nephron, with the filtrate, that's outside of our body. Okay? So here's what we're packaging them as the chylomicron. I hate that I drew the thing that looks like this because it looks like a lipid bilayer. It is not. It's a combination of the phospholipids and the apoprotein B48. So together, the combination of the dietary lipids and the apoprotein B48, that structure is an apolipoprotein. Okay? As are some other ones that we'll see in a little bit. So that's what we have for a chylomicron, exocytosed into the lymphatics via the intestinal lacteals to get them into the lymphatics, and then they eventually drain into the blood at the left subclavian vein. These are the only nutrients that don't pass through the hepatic portal system. So the liver gets first shot at all of your carbohydrates, all of your amino acids that you ingested, and also the short-chain fatty acids. But the rest of it, the bulk of your dietary lipids, are distributed to the body, throughout the body before the liver gets first shot. Okay, well, where are we trying to get these lipids to? Focus on the triglycerides. When we dump the chylomicrons into the blood, they're carrying primarily triglycerides, which represents work transporting fatty acids to the cells throughout the body. Well, what cells are particularly interested in them? The answer is most cells. Skeletal muscle works well. It has a lot of beta oxidation that can take place. Adipose tissue. Well, if we just ate and times are really good, we ate a lot of lipid, then this is a time for storing that lipid away for when times are bad. So adipocytes are going to take fatty acids and store them as triglycerides. Heart, again, great beta oxidation. Heart probably prefers fatty acids to any other foodstuff. Same for lung, kidney, liver. And kind of the one major thing that's missing from this list is the brain. Okay, but we're going to fuel much of the body, many of the cells of the body, based off of fatty acid metabolism. Okay, next trick. We've got a chylomicron. We made it so it could circulate in plasma by putting that coating of apoprotein B48 around the dietary lipid. How do we get those lipids to the cells that want to use it or can use it? And what we have to do again is break them down as they circulate in the blood, break them down into smaller fragments that can actually get into those target cells. And the way we break them down is using an enzyme called lipoprotein lipase. Got all sorts of lipases we're hearing about today. Lipoprotein lipase is an enzyme we find in the blood. It's attached to the endothelium. It wasn't made by the endothelium. It was made and secreted by adipocytes and skeletal muscle cells. So those two cell types primarily are producing lipoprotein lipase, releasing it out of the cells that made them, and then it becomes bound to the endothelium. Now, where in the circulation would you expect to see the highest concentrations of lipoprotein lipase then?
At the start,
At the start?
the below.
No, it's being made by skeletal and adipose cells.
I said at the start.
It's going to be primarily in blood vessels, in perfusing skeletal muscle and adipocyte. Right? That's where we released it. That's where it's got the greatest chance of binding when it gets into the endothelium, into the circulation. So we find the highest concentrations in those two areas, which means they're going to have the first shot at getting these dietary fatty acids that are produced. Adipocytes to store for the future. Skeletal muscle, we got a lot of skeletal muscle. It's going to be consuming a lot of these fatty acids. So what does lipoprotein lipase do? As a chylomicron, this kind of big structure, is lumbering through in our plasma, as it circulates, it ends up bumping into the walls of the blood vessels. And in that momentary time period, when it bumps into the endothelium, there's just that moment where the lipoprotein lipase can start tearing apart the triglycerides, just clipping off fatty acids. And so as the chylomicron continues to circulate around, every time it bumps into the wall of a vessel, and there's lipoprotein lipase there, a little bit more gets chewed up in terms of the triglycerides that are inside the chylomicron. And the product of the products of the lipoprotein lipase over time is it takes a triglyceride and it clips off all three of the fatty acids over time. Eventually we'll end up with glycerol and three free fatty acids. Now the glycerol, now let's do the fatty acids first. Picture yourself as a fatty acid. You can't swim well. Don't like an aqueous environment. It takes you to a higher energy state. And picture being panicked. Okay, so the free fatty acids, as they get clipped free, they diffuse out of the chylomicron, and chances are they're regretting it pretty much immediately. So what are they going to do? Two primary possibilities. One, they diffuse out of the blood entirely as quickly as possible. And so when they do that, the leading chances are they either enter a skeletal muscle cell or they enter an adipocyte. Either way, we're going to trap them once they're in there. Skeletal muscle cell will primarily run them through beta oxidation to fuel the TCA cycle. The adipocyte trap the fatty acid that it just got and then construct a new triglyceride inside the adipocyte to be used at a later time. The other possibility isn't that the fatty acid got immediately out of the circulation and into a nearby cell. but it stayed in the circulation momentarily or for some period of time by binding to albumin. This isn't a covalent bond, okay, but it's a temporary bond. It helps to get the fatty acid downstream and distributed further through the circulation. So those are the two primary fates, potential fates for fatty acids. The other product of lipoprotein lipase was the glycerol phosphate, or glycerol, sorry, not making it, I'm using it, is the glycerol. Glycerol is a 3-carbon sugar alcohol. It is hydrophilic. It can circulate in the blood, and where I'd like you to picture the primary endpoint for that glycerol is to be taken up by the liver. The liver can use it can phosphorylate it, and use it to make liver-derived triglyceride. Right.
Okay, so if it either diffuses out of circulation, or it's remade into something else, how do you end up with fatty deposits in like atherosclerosis, or because if it's leaving?
Those would be primarily, yeah, fatty acids that can get deposited. That's a lot of cholesterol too. Cholesterol would come through.
Okay, so that's a different thing. That these are not going to attach to the wall.
Well, you see, I avoid saying not. Always, words like always and never, I pretty much try to avoid. But when I think of plaque, I think more of cholesterol and another structure. Okay, and there we have glycerol headed to the liver for glycolysis, gluconeogenesis, whatever the liver may want to do with it. Free fatty acids either going to neighboring cells or binding to albumin. Okay, now that's how we're distributing dietary lipids. There's one other component we need to talk about, and that's if you look in the blood, not all of the triglycerides you find in the blood came directly from your diet. Because the liver has the capacity to make fatty acids and then combine them in the form of triglycerides. And the liver then can distribute these through, seems awful lot, can distribute the dietary derived triglycerides throughout the body. We'll take a break here, and when we come back, we'll look at how that feeds in. And we'll be talking about VLDLs, LDLs, and HDLs. And are different colors that might be handy to use here, or you can do it electronically, and you can annotate the slides on your computer. Use kind of, again, a couple of different colors. So this is showing me most of what is happening with lipid distribution. So let's start up on the upper left-hand corner where we have the small intestine. And here we're just going to see, we're going to review what we talked about with dietary lipids. It's the small intestine. And what I can see is you could draw in the lumen of the small intestine, you could draw a micelle. Or next to it, draw a micelle. And the micelle would be some circular thing like these other blobs. It would have a big TG and a little c. For a lot of triglyceride and a little cholesterol. And then on the outside, bile salt. It should be in the lumen of the small intestine, not inside the body. Now what we have on this diagram are the cells of the small intestine. And what we say, what they have labeled a nascent chylomicron. This is a newborn chylomicron. And what it has is a lot of triglyceride compared to cholesterol ester. So their shorthand says TG is greater than CE. When I'm drawing it out, I just use a big T and a big G and a little c. Okay? And then on the outside, they've labeled it ApoB48. I can recognize that. Somebody describes it, somebody draws it up. A lot of triglyceride, a little cholesterol, and apoprotein B48 chylomicron. And that's what got dumped first into the lymphatic system, and then from there into the blood. Now they've got more than what we talked about before, so we got to add a little bit to the story. Remember I said the chylomicron was made by enterocytes, first into the lymphatics and from there into the blood. Now what they're showing here, look at this diagram in the small intestine, where they said a nascent chylomicron, newborn chylomicron. And it's got triglyceride greater than cholesterol ester and apoprotein B48. Now here is that chylomicron, but now it's in the blood. And if I look at it, I see triglyceride is greater than cholesterol, right? This is still labeled a chylomicron. This one is called the nascent chylomicron. And here's my apoprotein B48. But how is this changed from when it was born to as it's circulating? I see two new apoproteins have made their appearance. Apoprotein C2, Roman numeral 2, and apoprotein E. Okay? So, if on a test or somebody's talking and they say, hey, I've got a structure and it's got apoprotein B48, the first thing I'm going to do is say, is it a chylomicron? They might say, well, kind of. They say, well, it's more triglyceride than cholesterol. Still a chylomicron. And then if they tell me, well, there's also apoprotein C2 and apoprotein E, I'll say, that's a mature chylomicron. So the difference between what they have labeled here as a chylomicron and a nascent chylomicron was suddenly these other two apoproteins have appeared. And we see that in the circulation. So where did they come from? They weren't born with apoprotein C2 or E. Where did they come from? And the answer is it came from high-density lipoproteins. Huh? Never mentioned that before, Tim. Well, what's a high-density lipoprotein? An HDL. Now, what's the other name that HDLs get labeled as? Good cholesterol. And cholesterol is cholesterol. This gets called good cholesterol. What gets called bad cholesterol? So we got to talk about that. So an HDL is a structure. It's a lipoprotein structure we find in the blood. It's produced by the liver. The liver makes these HDLs, or high-density lipoproteins. They're not as big and lumbering as a chylomicron would be. They're more maneuverable. And if we looked at an HDL, how could I tell an HDL? It contains cholesterol. It's born from the liver. It contains cholesterol we get out of the liver. And if we look at what's coating this cholesterol, it's apoprotein C2 and apoprotein E. So if somebody says, what kind of a structure has ApoC2 and ApoE only, and cholesterol, we'd say an HDL. Now the HDL has two primary functions. Function number one is to temporarily donate apoprotein C2 and apoprotein E to structures like chylomicrons. So where did, in the mature chylomicron, where did C2 and E come from? Came from bumping into an HDL as the two were circulating in the blood. And when that bumping occurred, some of the ApoC2s and some of the ApoEs transferred from the outer portion of the HDL onto the outer portion of the chylomicron.
Does the HDL also attain some of the equilibrium for you?
It can, yeah, it just transfers a little bit. Okay? So function number one for HDLs would be to be a donor of C2 and E, ApoC2 and ApoE, to things like chylomicrons. The second function of HDLs is as they circulate, they pick up, they grab free cholesterol. So they are born with cholesterol, and as they circulate, they start to accumulate more and more cholesterol. Cholesterol that, if it was left to just be free in the circulation, might start to form plaque. And therefore, that's why they get called good cholesterol.
Okay, but cholesterol is hydrophobic, right? What's the difference flow around in circulation anyway?
Well, when a cell dies, there's cholesterol in its membrane, and it just loses its home.
Yeah.
And there's another place that cholesterol comes from in the circulation. We'll talk about that as we go. Okay, so now I know the source of the C2 and the E. When I saw that mature chylomicron, the next question I might add is, well, why is that important? I mean, sure, I'm a teacher, so maybe I just tell you because I know it, and therefore I'm going to make sure you have to know it. But chances are there's a function for these two. The function for apoprotein C2 is it activates lipoprotein lipase. Now put that together. Think about it for a moment. When a chylomicron is born, it doesn't have any C2. Once it gets into the blood, we add some apoprotein C2. What's apoprotein C2's job? To activate lipoprotein lipase. And what's the job of lipoprotein lipase? To break down triglycerides in the chylomicron. So if we didn't transfer some apoprotein C2 onto the outer layer or level of this chylomicron, we would not be able to distribute the fatty acids throughout the body as quickly or efficiently as we do now. Okay? So we add the apoprotein C2 to increase or enhance our ability to break down the triglycerides that we had gotten from our diet. Okay, that's C2. What about E? Apoprotein E, what I want you to think of for its function is the liver has receptors for apoprotein E. The liver can recognize structures that have apoprotein E on their outer coating. And once they come in contact with the receptors on the liver for the apoprotein E, that structure gets pulled into the liver and digested. Well, how does that fit in with everything? We had a chylomicron born from an enterocyte through the lymphatics, and when it was born, it just had apoprotein B48, a bunch of triglycerides, and some cholesterol, all from the diet. It then bumps into an HDL as it's enters into the blood, and the HDL activates it. It makes it a mature chylomicron, because now the apoprotein C2 activates the lipoprotein lipase, and as the chylomicron goes through, the lipoprotein lipase molecules are digesting or breaking, I don't want to use digesting, breaking down the triglycerides and getting the fatty acids distributed to the tissues. And so as this mature chylomicron circulates through the blood, over time, it's getting smaller and smaller, because 90% of the lipid that started out in that chylomicron were triglycerides, And after a while, they're gone, or they're almost gone. So now we have something that looks like a chylomicron. If I was describing it would sound just like I described the chylomicron before, a mature one. Then it has apoprotein B48, C2 and E. But now I'm going to say, when I look at the inside, it has a lot of cholesterol relative to the triglyceride. We didn't add cholesterol, we just got rid of the triglyceride. So the only difference between this one and the one that was born or mature is now I see cholesterol is greater than triglyceride. The chylomicron has served its purpose. It has distributed dietary triglycerides to the rest of the body. Now it's no longer a nascent chylomicron or a mature chylomicron. It's a remnant of itself, and that's what it's called, a chylomicron remnant. You can tell the difference between a chylomicron remnant and a mature chylomicron because in the remnant, cholesterol is greater than triglycerides. And at that point, here was my HDL, born from the liver. It's donated its apoprotein C2 and E to the chylomicron. And now, our chylomicron has been broken down as it circulated through the blood by lipoprotein lipase. And now we have a chylomicron remnant, where we have apoprotein B48. Now when we get to the remnant part, where cholesterol is greater than triglyceride, we donate the apoprotein C2 back to the HDL. It served its purpose. We've gotten rid of almost all our triglyceride. But we keep the apoprotein E. Why? Because it can bind and be recognized and bound to receptors for apoprotein E in the liver. And so the liver endocytoses the chylomicron remnant, which now primarily contains dietary cholesterol. And that's where your dietary cholesterol is going to, primarily to the liver, being contained in the chylomicron remnant. So the liver gets most of the cholesterol you ingest. Okay? Now we saw that the HDL was made by the liver, donated apoprotein C2 and E, scavenged cholesterol, and eventually it was born from the liver, it goes back to the liver to be endocytosed and destroyed, digested. One other player, actually two forms of one other player, is that you have to recognize that not only can you eat triglycerides and have to distribute them through the body, but it's another function of the liver. Remember, when in doubt, if you don't know anything, you're asked, where in the body does this happen? Say liver. Another function of the liver is liver can make its own fatty acids. And it can then take those fatty acids and turn them into triglycerides. And the liver can then package those triglycerides up and send them off to the rest of the body. Well, how is a liver cell in the hepatocyte going to make fatty acids?
It pulled from the adipocytes?
No, it's going to make brand new fatty acids. Never before seen. De novo synthesis of fatty acid. Something like palmitic acid, palmitate, 16 carbon saturated fatty acid. I got to get my hands on some carbon in order to make them. Huh? Glucose. Damn it. You can eat sugar and get fat. Right? I told you before. Done the experiment over and over and over again. It always comes out the same way, but I think maybe this time it's going to be different. It never is. You can eat sugar and make fat. Now, it's kind of unfair. You can't then break down fat and make sugar from it. So fatty acids basically are no good for gluconeogenesis, only off-chain fatty acids. Okay? So the liver is where this alchemy occurs. The liver is what's taking glucose. So remember the hepatic portal system? The liver gets first shot at all that glucose. And the liver's got glucokinase, which when there's a lot of sugar around, a lot of glucose in the blood, the liver is going to be able to trap it better than any other place. And what's the liver going to do with all that? It's going to make some glycogen, but it's also going to start making a lot of fatty acids. So now the liver's accumulating fatty acids. What's it going to do? Well, take a little offshoot from glycolysis and get glycerol 3-phosphate and use that as a backbone, and the liver makes some triglycerides. The liver also has a little bit of an issue with the tendency for cholesterol to accumulate. Your dietary cholesterol is primarily coming to the liver. Just mentioned that HDLs scavenge cholesterol from things like dying cells. And those HDLs return to the liver with even more cholesterol than they started with. The liver's got that. And then number 3, strike 3, is the liver can synthesize cholesterol. Okay? It's not just dietary. The liver can synthesize. So now the liver's a repository of cholesterol, has the ability to make triglycerides. And so what the liver does is say, I'm going to take a bunch of triglycerides. Let's see if I can get rid of some of this cholesterol. I just picture it kind of, well, doing the body a favor. Maybe it can stick a little bit of cholesterol in there and nobody will know the difference because I'm drowning in this thing. And it needs to package it up. And we saw how we could package these lipids when we talked about dietary lipids. We need an apoprotein. In the case of the liver, it makes something called apoprotein B100, which is almost exactly double apoprotein B48's number. That's not surprising how it got its name because it's almost like a dimer of apoprotein B48. The main thing you need to know is if you see apoprotein B100, that thing came from the liver. If you see apoprotein B48, it came from an enterocyte. Okay? Now, the liver doesn't fool around. When it packages these things up, all these triglycerides and some of the cholesterol, it even puts chylomicrons to shame. It produces really big, lumbering structures, and those are called very low density lipoproteins, or VLDLs. And you can tell a VLDL from its description because it'll contain apoprotein B100, a lot of triglyceride, and a little cholesterol. And just like we saw for a chylomicron, it matures. At birth, that's all it had was B100, a lot of triglycerides, and a little cholesterol. But as it circulates, it too bumps into HDLs, and it gets activated with apoprotein C2. And it receives those, the apoprotein E, the liver will recognize eventually and take it back up and destroy it. So now these big VLDLs are lumbering through the blood. They're activated with their apoprotein C2, and they activate lipoprotein lipase, and over time, the VLDLs start to shrink as they lose their triglycerides. And eventually we get down to the point where most of the triglycerides, and remember these are liver-derived triglycerides, most of those triglycerides are gone. So now we have the equivalent of a chylomicron remnant, but it started out as a VLDL, a very low density lipoprotein. What we have now is called a low density lipoprotein, or LDL. I know when I have an LDL because it's got apoprotein B100, it's got C2, it's got E, and its cholesterol is greater than its triglyceride. That's how I can tell the difference between an LDL and a VLDL. And the LDL gets called bad cholesterol. I said cholesterol's cholesterol. Why is this bad? Because it's leaky. LDLs are the exon Valdez's of lipid transport. Okay? They leave a little cholesterol slick as they circulate around. And that can start to tie up and start to bind to the lumen of your blood vessels. You can build up plaque. Now the HDLs are going around saying, what the hell are you doing? What were you thinking, trying to collect? And also scavenging cholesterol released from dying cells. So that's why LDLs get called bad cholesterol. Yeah,
Why are they so leaky though?
that part I don't know. Just that it doesn't do a real good job. It may have been secret instructions from the liver saying, I'm trying to get rid of this cholesterol. You know, it wouldn't bother me if you didn't come back with it. But I haven't been able to find the code for that. Yeah.
Is ApoB 100 made specifically for VLDLs and LDLs? Or is it?
Right. If you see B100, it came from the liver and it originally was in a VLDL.
Is it reasonable to presume that someone with liver failure would have atherosclerosis since they can't process that cholesterol, or no?
It wouldn't surprise me, I guess. I'm thinking off the top of my head, but there are some other problems that are coming up with liver disease. But yeah, the liver isn't able to contain and try and, what's the one way we can get rid of cholesterol from our body?
Fiber.
Hmm?
I was gonna say fiber, but.
Well, fiber will help us, but how does, fiber is carbohydrate. What's that got to do with cholesterol?
Does not hold, form a bowl.
It ties up cholesterol, but where was the cholesterol? Where do you find the fiber?
The lumen of the gut.
In the lumen of the digestive tract, which is outside of our body. So how are we going to find cholesterol to tie up there? You know the answer. Well, dietary is going to reduce the amount of cholesterol we absorb by having more fiber. But I'm saying we can actually get rid of some cholesterol, also by using fiber. Cholesterol that was already in our body, how can we get rid of that using fiber? Two-word term, that's all I want to hear. Can't even think of 2 words.
So we would need some enzyme that pulls cholesterol out of the bloodstream and into the.
Nope, nope. I'm already doing it. I told you the answer earlier. How could you get cholesterol into, cholesterol that's already been in your body, how could you get it into the lumen of your digestive tract?
Break down something?
Bile salts.
Wait, those only work, but those are dumped into the lumen, right?
Those are produced by the liver, dumped outside of the body, and then we get 95% recycling. But if we have fiber, just like it could do to the dietary cholesterol, it could start binding up some of those to reduce the recycling rate, in which case we should start to lower our cholesterol.
Well, I just don't understand how that pulls it out of circulation into the lumen of the gut.
Because the liver is producing the bile salts and dumping it into the bile. That was the last really cool drawing that I made the other day, the other lecture. The liver, the hepatocytes are combining cholesterol, making it into cholate or chenodeoxycholate, and then adding the glycine and chlorine to make glycocholate and chlorocolate, and throwing that out of the body.
Can you explain it one more time, so we can,
Yeah, I'm aware of all that stuff going, so you've eaten a meal, and it's traveling along, dumps emulsifier in, that's fine, but you're saying it's pulling it from the blood and then putting it into
Picture eating regular food but no fiber. Eat fat but no fiber. Your liver dumps out 100 bile salt molecules. Of those 100 bile salt molecules, they're outside of your body now. However, later on in your digestive tract, 95 of them are brought back into the body and given to the liver to recycle. Okay? That was when you ate just fat, no fiber. Now, we're going to have you eat fiber along with the fat. This time when you eat the fat, but with the fiber, once again, your liver produces 100 bile salt molecules and dumps it into the lumen of the digestive tract. Emulsify the fat, we absorb the fat, but now, out of those 100 bile salt molecules, 20 of them end up binding to the fiber. And now only 80 of the molecules get brought back into the body and be recycled. So in that example, we lost 15% more bile, 15% more cholesterol due to the presence of the fiber. I just picked numbers.
Because the bile isn't necessarily attached to cholesterol.
The bile salts are made from cholesterol. Okay? So the VLDLs become LDLs, and the LDLs are leaving this cholesterol oil slick behind them, but eventually they're taken up by the liver, broken down, and the pieces reused. So that's the whole system, and you can, if you look at it, you could start up with the intestine, go along through here, down through here, back to the liver, then produce a VLDL. Once again, bump into an HDL. The VLDL comes through, we distribute the fatty acids from the VLDL, eventually causing it to become an LDL. And some of these go to extrahepatic tissue, but I want you just picturing they're going to come back to the liver. There's more than these. There's an intermediate density lipoprotein. Don't need to worry about that. And you don't need to worry about this page either. You just scribble it out. The only thing here, primary functions, chylomicron, donate, I would put in the word dietary free fatty acids to tissue. VLDL, donate or deliver liver-derived, I've got it here, deliver triglycerides from the liver to tissues. Actually, it's delivering the free fatty acids again. LDL, I don't like what I have here. It says it's LDL, the liver cholesterol to tissues. That's not why an LDL, LDLs aren't made. They're the remnant of a VLDL, and the VLDL was made to distribute fatty acids to the body. However, I put this down here because this is what happened. We see cholesterol from the liver leaking out of LDLs going to the tissues of the body. Now, it serves some purpose to have enough cholesterol in membranes. But the main reason I put that in here was I saw a released dental board question that asked, what's the function of LDL? And what they have keyed as the correct answer is to deliver cholesterol from the liver to the tissues of the body. I may not agree with it. It was the only potential answer that worked there. But somebody somewhere else may write a question about LDLs and say, yeah, they're there to deliver cholesterol to the tissues of the body. HDLs, I said, it's a reservoir for apoprotein C2 and E, that the C2 activates not just chylomicrons, but also VLDL. Okay? And because it stimulates or activates lipoprotein lipase. And the HDL scavenge cholesterol. What you don't need to know here is this detail that goes on. So ignore that part. There's a dying cell. HDL's picking up the cholesterol. Okay? And I think that's it. Does that make sense? Did you know you could fit lipoprotein in so many ways in so many different structures? Do you know how to identify the different structures? Because that's an easy question to write for a test. As I get older, I could call it easier, or we'll just be honest, I get lazier.
What do you mean by identify different structures, like?
If I give you, what would be a possible correct answer if I told you, here's a structure that contains apoprotein C2? What could be a possible correct answer?
HDL.
An HDL.
LDL.
LDL. Huh? Mature chylomicron. A VLDL. Okay? Just saying there's apoprotein C2. Everything we mentioned has C2. What if I said it contained apoprotein C2 and cholesterol is greater than triglyceride?
LDL.
Could be an LDL. Could be, no.
Remnant?
Could be, well, what's a remnant of a VLDL?
Intermediate
An
LDL.
LDL. So now there's a structure that has apoprotein C2 and also cholesterol is greater than triglyceride. There are three possible correct answers.
Chylomicron, mature chylomicron.
Huh?
Mature chylomicron.
No. Mature chylomicron triglyceride would be greater than cholesterol. Chylomicron remnant, yeah. So now I've got LDL, chylomicron remnant. One more. C2, and then more cholesterol than triglyceride. HDL. It's born with a bunch of cholesterol, not much triglyceride at all. Okay, one last quiz. What if I said, I have a new question, what would be the possible correct answers? If I said, here's a structure that contains apoprotein B100.
Say that again.
VLDL? Anything else? And an LDL. What structure contains no apoprotein? Micelle. Ready for the toughest one? What structure in the body contains no apoprotein?
Muscle cells?
Yeah, none of the structures inside the body are lacking apoproteins. Everything that was in the body contained apoprotein. Outside the body, we had a micelle and it used bile salt for the comparable function.
Is the brain included as well?
Brain included for what? For parties, you should include it. Cerebellum likes to party more than any other part of the brain. That's why you find it difficult to do that when a police officer is.
But is the apoprotein part of that?
Apoproteins we find in chylomicrons, chylomicron remnants, HDLs, VLDLs, and LDLs, but not in micelles. Okay? Well, my wife's supposed to be out of the recovery room, so I will set you free and I will go back to pretend I've been there the whole time. I couldn't leave you there. No, the sad part, and this is, right next to the hospital, she's at Euclid General, right next to it is the Hospice of the Western Reserve.